So thank you, Peter, for that introduction. I believe you have given half of my talk already, and I think the other organizers, this has been a great experience for us, and I'm really impressed to see how many people are engaged in supporting education in science in Hungary. So I was asked to provide a bit of background, how I became interested in science, and a little bit of background about my research. So I'm going to first talk briefly about my career path in terms of education, and that led to me becoming an independent investigator. And then I want to briefly summarize what the field of my research has been, G protein-coupled receptors. And then finally, a brief history of the GPCR field and my contributions to the field. So, as Peter mentioned, I was born in Little Falls, Minnesota, which was famous because it was the boyhood home of Charles Lindbergh. I'm sure most of you don't know who Charles Lindbergh was, but he was the first to fly across the Atlantic from United States to France. The town was really small. Here is our Little Falls, which they turned into a electric power dam. And again, my family, my father, who owned and operated the bakery, my mother, my sister, and myself. At some point, we all worked in the bakery. My mother was a cake decorator. My sister sold the goods at the counter, and I did a number of odd jobs, including slicing bread. Here's just some pictures advertising our bakery, which said we could send decorated cakes anywhere in the country. I don't think we ever sold a cake any place outside of Little Falls, but we had ambitions. So my town was relatively small, and you can see sort of the major points of interest for me. My home was on the north end of town. I was born in St. Gabriel's Hospital on the south end, my high school, and here is the home of Charles Lindbergh. So as a boy in Little Falls, as Peter mentioned, I became interested in cycling, and I show this picture again because here I am at 14, and you can see I looked probably like I was 7, and my friend, who is 3 years older, wasn't all that much bigger, and we went cycling to Yellowstone National Park. And I particularly want to point out to Tim Hansen because his father was a pediatrician. They lived about 2 blocks from our house. And I think I became interested in medicine because I really admired his father. He would help. I remember being in their home when people would bring their sick children to his home, and he would take care of them. So I think that's where I first became interested in becoming a physician. I also, additional bicycle trips included a trip across the country, and then this one is particularly interesting for me. So this one was a trip that I took in 2005 with my son. It was the first bicycle trip that we took together, and we were touring the Pyrenees during the Tour de France. And this was also particularly notable because Tungsten and I, my wife, had just come from the synchrotron in Grenoble, where we found the very first evidence that we might be making some progress in our project. So from Little Falls, I went to the University of Minnesota in Duluth, and some really important events happened. I met my wife the first week I was there, and we've sort of been together ever since. And my first scientific mentor, Conrad Furling, was a biology professor, and he ran a small lab. There were no graduate students in the program, so he had to recruit undergraduates. And my research project involved developing culture for this worm called Chironomus tentans. they had the salivary glands that had polytene chromosomes. And that means they were replicated many times, and you could actually look at chromosomal activity using a light microscope. So this was my first publication with Professor Furling, where we developed a way of maintaining these salivary glands in culture for periods of time. So this gave me my first experience in research. It was fairly descriptive. It wasn't enough to change my mind from going to a physician to a basic scientist. And I subsequently went to Yale University. Another important event in my life is I married Tang Sun after my first year in medical school. And I also had a research experience. We were required at Yale to have an MD thesis, which was a relatively small research project. And I ended up studying the genetic basis of rotavirus. So rotavirus is a very common virus that causes diarrhea in children, and it rapidly evolves because of the special genomic structure that it has. After medical school, again, I found this of interest, but somewhat descriptive, and it really didn't change my mind in terms of becoming a scientist or a physician. And I subsequently went to St. Louis, Missouri, Washington University, where I became a resident in internal medicine. And at the time, my son was born a week after we arrived. So my poor wife was pretty much alone with the child while I was on call either every other night or every third night. So my son Jason was born in 1981, and my daughter Megan was born in 1983. It was also here that I began to be interested in the field of intensive care medicine. So of all the rotations I did, I really enjoyed being in a cardiac unit or a pulmonary acute unit, primarily because you could see patients getting better or worse in a relatively short period of time. You would make some intervention, and you could see whether it worked or didn't work, and you could change the intervention. So it was in a way like a physiology lab where you were really fighting to make sure your patients got better. And it was during this time that I decided that I wanted to specialize in intensive care medicine. So I chose, at the time, you either became a pulmonologist or a cardiologist. So I chose to become a cardiologist and looked around, and I was fortunate to be able to get into the program at Duke University. Now, Duke was one of the largest training programs for cardiologists in the country, and they were particularly interested in fostering research among their fellows. So as a cardiology fellow, I could begin in a lab and then do my clinical rotations, or I could start in clinical rotations and then find a lab. And I had 2 young children and was tired of being on call, so I chose to start in the lab. And I looked around, and I found the lab of Robert Lefkowitz. This was a rather sort of unsettling decision because there were a number of other great labs that were much smaller. At the time, Bob's lab was about 25 people with only 2 graduate students, and it was a very intense place. But he was working on something that I thought was close to my clinical interest, and that is adrenergic receptors. And I'll explain that in a little later. So here's a photo of my young family in a very beautiful area of the city where we were able to live, Jason and Megan. So at the end of my training, and I'll spend some time here telling you about what I did at Duke and how it led to my interest in furthering this career as a faculty member at Stanford. So I joined Duke in 1984 with the idea of becoming a cardiologist. I started in the lab, and when things started working, I kept putting off my clinical training, and I put it off to the point where I no longer was capable of becoming a cardiologist, and I gave up the idea of intensive care medicine and moved to Stanford with the idea of being an internist and hopefully a researcher. So when I joined Stanford at the beginning of 1990, I was allowed 25% of my time for research and the remaining time for patient care, which included attending on the medical service and a substantial amount of moonlighting to help facilitate financial issues in the family. We had a big mortgage, and at the time, my wife, Tang San, started medical school. So this is sort of the trajectory. I'm still at Stanford, and I'd now like to tell you a little bit about the field that I became interested in at Duke and some of the progress that we've made over this time. So what are G protein-coupled receptors? So they are primarily important because they mediate the senses of the majority of, they mediate the response to the majority of hormones and neurotransmitters. And a model of how these receptors worked when I first joined the Lefkowitz lab is shown here. So we knew there were proteins in the plasma membrane. We knew that when activated, they engaged heterotrimeric G proteins. These consisted of alpha and beta and gamma subunits. And then when the receptor activated the G protein, guanine nucleotide GDP was released and GTP bound. The G protein would split into an alpha and a beta gamma subunit, and these would go and then modify the behavior of other enzymes or channels in the cell until GTP is hydrolyzed, the components would reassemble, and the cycle would begin again. So our goal at the time, when I was in the Lefkowitz lab, was to begin to understand how these worked, initially by cloning them and determining their primary structure. So in addition to their role in hormones and neurotransmitters, they also mediate the senses of sight, smell, and taste. And we now know that there are roughly 800 members of this very large family, including roughly half of these include receptors for olfaction, 4 for vision. Most of my research has been in a subfamily called family A on adrenergic and muscarinic receptors, as well as opiate and cannabinoid receptors. We've also done work in family B, and you'll probably recognize the GLP-1 receptor, which is a now very popular target for both diabetes and obesity, and family A, an important target for neurotransmitters. But my real interest started because of my interest in the autonomic nervous system. So as an intensive care doctor, you're often giving patients drugs that interact with adrenergic receptors or muscarinic receptors. So the autonomic nervous system is important to prepare us in times of stress. So the fight or flight response increases heart rate, increases blood pressure, and it shunts blood away from viscera towards skeletal muscle to prepare you to flee from predators. And the opposite system is the muscarinic system, also called the rest and digest system, which slows down heart rate, improves the mechanism of the gastrointestinal system and the bladder as well. So I was particularly interested because of the role these play in the heart. And as I mentioned, in the intensive care unit, we are often giving patients drugs that would improve their heart function, their heart rate or contractility, would improve blood pressure, either, and sometimes it would mean increasing their blood pressure or reducing their blood pressure. And here's just an example of a really interesting circuit. The heart, the vessels around the heart have special sensors which detect your blood pressure. It's either too low or too high. That sends information to the midbrain, which then tells the system either to increase sympathetic tone or increase parasympathetic tone. And here's just a blow-up of a nerve innervating the heart cell and releasing adrenaline onto beta receptors in the heart. So this was a scheme that was pretty well understood at the time that I joined the Lefkowitz lab, and something that I was really interested in understanding the principles behind these receptors, how they worked, and how they were important in engaging the G proteins. So as Peter mentioned, these are really important drug targets, and as shown here, they're primarily responsible for the majority of pharmaceuticals on the market. Roughly a third of all marketed drugs today work on G protein coupled receptors. So I'd now like to just give you sort of a brief history of advances in the field. The field really began in the early 1970s with work from Marty Rodbell, who identified G proteins as being important signaling molecules in transmitting information from the plasma membrane to the rest of the cell. G proteins were demonstrated biochemically by Ross and Gilman in 1977, and Gilman and Rodbell would ultimately win a Nobel Prize for their work in G proteins. A major advance, and I'll talk a little bit about this in a minute, is in 1984, the first beta adrenergic receptor was purified from a native source, and that gave us the first tool that we needed to do the cloning, which subsequently came in 1986. So I joined the lab in 1984 at this really sort of auspicious time when really important things were happening, giving us the tools ultimately to be successful in cloning. And so I'd just like to briefly mention this. So this is Jeff Benovic, who was a graduate student at the time that I joined the Lefkowitz lab, and he had worked for a number of years to be able to purify beta-2 adrenergic receptor, receptor that's in the lungs. So he achieved a 33,000-fold purification to obtain a tiny amount of receptor from lungs from roughly 1,000 hamsters. So this is a huge effort. He then took that beautiful preparation of protein and digested it with cyanogen bromide, a protease, and obtained a number of different peptides. And from these peptides, we could guess the nucleotide sequence that was necessary to make these peptides. And these guessed oligonucleotides were then used to clone the receptor. And to make a very long story short, we were able to obtain the clone using this particular guest probe from a genomic library. And we were very lucky because the beta-2 receptor has no introns. And that meant the entire coding sequence from the receptor could be obtained from a genomic clone. And here's the primary amino acid sequence. And what we noticed is that there were 7 stretches of hydrophobic amino acids, suggesting that this didn't just span the membrane once, but spanned the membrane 7 times. So this was our first clue. The secondary structure of the beta-2 adrenergic receptor was our first clue as to what these receptors look like in 2 dimensions. And it was the first time that I really thought that I'd love to be able to see what this looked like in 3 dimensions. And here's my mentor, Bob Lefkowitz. After the cloning, it was a very big event in his career, and he went out and bought a Porsche and had what we call vanity plates, Turbo Bob. So another memorable experiment when I was in the Lefkowitz lab was beginning to try to understand what parts of this protein dictated ligand binding and what parts dictated interactions with G proteins. And part of the goal was to be able to obtain other clones so that we could do chimeric receptor experiments. And I also collaborated with John Reagan, who was another tour de force. He was able to purify alpha-2 adrenergic receptor from human platelets. And I'll note that this was a time when AIDS was just becoming prevalent, and he was exposing himself probably to HIV virus for a number of years before he ultimately got the purified protein. And in this experiment, which was one of my favorite, Tungsten and I worked very closely to do this experiment, We created a number of chimeric receptors consisted of parts of the alpha-2 receptor and parts of the beta-2 receptor. And then we could characterize their binding properties and their G protein coupling properties. And we found a few that coupled like an alpha-2 receptor but had the pharmacologic properties of a beta receptor and conversely. And this gave us the first clue that the G protein coupling domain was in this large third loop, and binding domains were particularly important in the transmembrane segments. So from my time in the Lefkowitz lab, in 1990, I landed a job at Stanford, and just briefly, as I said, my major goal at the time was to try to understand the structure of these various proteins in 1990. And it wasn't until 1995, and this was not work from my lab, that the structure of a G protein was first solved. And it took much longer until 2007, when we were finally able to get a structure of a receptor in the inactive state, and then ultimately structures of a receptor in active state and in the G protein coupled state. But as you noticed, I joined Stanford in 1990 with the intention of really obtaining these structures. But it wasn't until 2007, roughly 17 years, until we finally succeeded. And now I'd like to tell you briefly, explain why that took so long. So if you wanted to know the structure of a protein at the time, you needed to use a method called X-ray crystallography. So you would start with your protein, your purified protein. So you first of all need to be able to make a lot of protein. You would then try to coax it to form crystals. And these crystals would look something like salt crystals that you use every day. You could then bring these salt crystals, or these protein crystals, to a synchrotron, an X-ray source, where you would shine X-rays into the protein crystal. The X-rays would diffract off the atoms, and these diffraction patterns would then allow you to calculate a structure, a three-dimensional structure. So our first goal, which again took much longer than I anticipated, was to be able to purify beta-2 receptor, get enough protein, milligram quantities, that would allow us to do structural biology. And we ended up using insect cells that we infected with a special kind of insect cell virus, and then worked out a purification scheme. And then finally, and this was around 1995, we were able to make enough pure protein that we could begin crystallography trials. But we also began other kinds of studies that ultimately helped us with crystallography. But in 1995, we were very excited. We could make enough protein, and we thought it would be just a matter of a few months, maybe a year or so, before we had our structure. But as hard as we tried to grow crystals, we could never get this protein to behave well. But as I mentioned, while we were failing to grow crystals, we were beginning to get other kinds of information about how this protein worked using biochemical and biophysical methods. And I'm just going to briefly summarize what we learned that was important. So early on in our studies on receptors and the mechanisms, the model was very simple. It was believed that a receptor existed in an inactive state and an active state when bound to an agonist. And that, so this is a simplified two-membrane protein, but it illustrates that we thought that we could simply stabilize the active state with an agonist, stabilize the inactive state with an antagonist, and then when the receptor was in the active state, it could engage the G protein. And this two-state model would suggest that, well, these are two stable states, they should be relatively easy to crystallize. But it turned out, based on our biophysical studies, this wasn't the case, and that the receptor actually behaved in a very different way. It was highly dynamic, in that even in the absence of any drugs, it would largely be in the inactive state, but occasionally could wander into an active state. So it was highly conformationally complex. when you bound an agonist, it would increase the probability that a receptor would be in an open state that could engage the G protein. But similarly, it was also in inactive states. And only when you had a receptor G protein complex did you actually stabilize a single state in the receptor. So this is a better kind of view, molecular dynamic simulations of what a receptor behaves like. It's highly dynamic. We found that particularly two domains, this transmembrane 6 and transmembrane 5, were the most dynamic. And again, agonists alone didn't fully stabilize the active state. So this was a particularly challenge, and why is that? Well, if we imagine the beta receptor, or any receptor, is conformationally uniform, in other words, it's not dynamic, it stays in the same conformation, then we can represent that as hexagons that would pack nicely into a two-dimensional crystal. And that's what we thought we were dealing with when we started. However, based on our biophysical studies, we knew that the receptor was conformationally heterogeneous, particularly this transmembrane 6. And that meant that even though we had pure protein, they had different shapes. And these different shapes wouldn't pack together nicely to form a nice crystal. And that was our major challenge. How do we get this to behave better so that we get a crystal? So our first problem was protein dynamics. And the other problem was that we were dealing with a membrane protein. And although we could purify the protein out of membranes using special detergents, most of the receptor was still covered with detergent molecules. And there were few places where the receptor could form what we call crystal lattice contacts. So parts of the protein that would engage other parts of the protein, another protein molecule, in forming a crystal lattice. And so we took two approaches. One was to create antibodies that bound to the most dynamic parts of the protein, and they would also provide polar surface. And the other was protein engineering. In other words, to try to engineer the protein so that it would behave better. And both of these methods worked pretty much the same time in 2006 and 2007. And our first crystals were of an antibody complex, and you can see these spots extend from the center of this the beam outward. And the further outward you see the spots, the better quality of the crystal and the better resolution. So this was a roughly 3.5 angstrom crystal, and it was our first crystal, but the subsequent crystal of a modified receptor, of an engineered receptor, was even better. And this made use of a special kind of environment called lipidic cubic phase. And you can see now we have really nice spots all the way out to 2.2 angstroms. So both of these methods worked. We had our first structures of the inactive states in 2007, and here's a receptor antibody complex, and here's a receptor T4 lysozyme complex. So we inserted T4 lysozyme between transmembrane 5 and 6. T4 lysozyme, it can crystallize in almost any condition. It's probably the most crystallized protein in the database, and it not only stabilized our receptor, but it provided extra surface for forming crystals. The next challenge was to get an active state. So we had an inactive state, and we tried to get an active state. We ultimately were able to get a receptor bound to an agonist, but it crystallized in the inactive state. So this was very disappointing, but we went back to our model of how this receptor worked, and we realized that even in the presence of an agonist, the receptor was largely in an inactive conformation, and we needed something to stabilize it. We were trying to get a receptor G protein complex, but that was failing. So we went to antibodies again, and I met a Jan Steert at a Gordon conference, and he suggested we try antibodies that we obtained from llamas. And these are special antibodies, unlike our antibodies where we have heavy and light chains. They're heavy chain only, and they only have one variable domain. And so these variable domains are particularly useful for fitting into crevices and stabilizing protein structures. So using our nanobody that we obtained from a llama, we were able to get our first active state structure. But later that year, we also succeeded in getting the receptor G protein complex. And I want to really emphasize that this was an incredibly collaborative effort that involved individuals who all contributed special techniques or reagents, including expertise in G proteins. We had a very high affinity agonists that were obtained from a pharmaceutical company, special detergents. We also had nanobodies that were involved. We did some protein engineering, and a lot of our work was guided by electron microscopy. And then we had a special lipid environment. So all of these were essential, and we were finally able to grow these crystals. And here are the major players coming back from the synchrotron after we collected our data. Roger Sunahara, Andy Cruz, my graduate student, Brian DeVries, Roger's student, and Soren Rasmussen, who's a postdoc who really did the majority of this work. So now we had all states, or all of the most important states, inactive, agonist bound, but not fully active, and then the fully active state. And I'm just going to briefly show what we learned from that. We could see the receptor going between inactive and active states and begin to understand the structural changes that occurred in the receptor upon agonist binding and conformational changes in the cytoplasmic surface. And there's a really elegant pathway we call allosteric pathway from small changes due to agonist binding that propagate into larger changes in the cytoplasmic surface that then allow the engagement of the heterotrimeric G protein. And we were fortunate that my wife Tang-Sun had planned a meeting, GPCR workshop in Hawaii, and the majority of my colleagues who were involved in this work joined us, and you can see it was a huge effort, huge collaborative effort, and I'm particularly grateful for being able to work with so many talented young people. And here's the day in 2012 when we were awoken at 2:30 in the morning, and the house began to fill up with people, and it was a very exciting morning learning that we had won the Nobel Prize. Again, my lab at the time, I had incredibly talented students and postdocs who all helped with this work. And my co-awardee, Robert Lefkowitz, it was great to share this prize with him and also to share it with my family. And I've been really grateful that through all my scientific career, I've had a very supportive wife, not only who supported me emotionally, but also supported me in the lab and worked together with me for many of my years in science. And I think I'll end here, and thank you for your attention.
So, Professor Brian Kobilka, thank you very much for your speak. I can see that I was learning about this in 2013 the first time, but a lot of things has been going on since then. So maybe I have to go back to med school and learn it again. So the floor is open to questions, please. Can they please take the mic and...
Congratulations, very nice talk. I would like to have a question about, do you have data about receptor phosphorylation? What do you think? Why do these new opioids do not induce arrestin interaction? Is it the absence of the core interaction or the absence of the tail interaction?
That's a really good question. And I don't actually know that we know that for sure. So there are basically two classes of, and this is kind of confusing, Class A and Class B, family A receptors. And Class B have a tight core interaction, and Class A, beta-2 receptor, and the opiate receptor don't have such a tight, so it's possible they're only interacting with the tail. But we don't know as much about that for the opiate receptor as we do for, say, the beta-2 adrenergic receptor.
Thank you very much.
Okay, next question, please.
Dr. Kobilka, that was great. Thank you very much. Can I ask, what is the prevalence of genetic variation amongst the GPCRs which could account for pharmacogenomic effects?
It's a great question, and I should know the answer, but I don't. But there's a researcher who's really been focused on this, Madan Babu, B-A-B-U, and he's done a lot of this with, including the opiate receptor. But I don't know. I do know that there are now it's now recognized that there are many more polymorphisms than previously appreciated.
Next question.
Wonderful talk, Brian. My question is that you mentioned that the respiratory depression is not RST related, and you were focusing on the GZ. But as far as I could judge on the figures, most of the things which did not cause respiratory depression also did not cause RST response. So basically, what is your argument behind this?
Yeah, so we don't, so none of these show respiratory depression. We now have one that does, and we need to, it actually leads to respiratory depression. And we're just now doing the panel. But I think we have to have more selectivity, and it's not likely to be just one. It's likely to be a combination of two different G proteins.
Please, any more questions?
So congratulations, that was really brilliant presentation. So actually I am thinking about the preclinical testing of the different drugs and these proteins. So in these receptors has a huge variability. So how can you be sure when you use a drug that which subtype of receptors are acting? So what are the models? Because of course in medicine we have plenty of knockout models and so on, but here there's this huge variety of these kind of receptors.
Yeah, so we have to worry about two things. One is which receptor are we using in our assays? And so far, my student has followed this, and we're using the most prevalent in our assays for structures and functional assays. But we still have to be concerned that, as I mentioned, some of these polymorphisms will have different signaling behavior. The other is that we test these drugs in mice first, and then other animals, and there are differences. For example, we also work on the cannabinoid receptors, and the CB1, which is a target for pain, is there are significant differences between mice and humans. So in that case, we're making humanized mice. In other words, we're knocking in the human receptor into the mouse genome so that they will express only human cannabinoid receptors. It turns out that the mouse and human opiate receptors in the binding pocket are pretty much identical. Nevertheless, they're different biochemically. So if you express a mouse receptor and a human receptor, purify them, you're going to get more functional mouse receptor. They tend to be more robust. So even though the binding pockets are the same, there are other properties that we might have to be concerned about in terms of using a mouse as a model for testing our drugs.
Sorry, I have one more question. Concerning, of course, these G protein coupled receptors are there for physiological reasons and not for pharmacological reasons. Of course, we are trying to interact with different drugs. But do you maybe for the future thinking about like the gene editing of the original receptor to have the physiological response more strong, or maybe to revise some pathophysiological events?
So I can't imagine how you would be able to edit genes for all receptors in the brain, for example. Yeah. How you would get the editing material to the different locations. Maybe you could edit an embryo, but I don't think that's going to happen anytime soon.
Yes, please.
So you mentioned actually, and your data clearly showed that there is very nice bias between the different signaling pathways, but bias has new dimensions, like temporal bias and also spatial bias. And probably a receptor is signaling differently from the plasma membrane and signaling from the inside. And I just wondered that, what is your opinion about this? Obviously, your approach is basically targeting the receptor, which is in the membrane.
Yeah.
But, so what do you think about the spatial aspect of this?
Yeah, so I think that's a really important new area of investigation in the GPCR field. And one of my colleagues, Ruth Huttenheim at Stanford, is actually actively looking into this. So what are the signaling proteins located adjacent to the receptor in the plasma membrane? What it internalizes, what signaling proteins are involved there? I don't know the answer yet, but she'll figure it out, I think.
Okay, any more questions? Maybe until then I would have a question, basically, because what we can see that a lot of people are doing also basic science, and they may have good results. However, they don't end up to bring this back to the bedside as well. But as we can see in your presentation as well, that you have a great network of scientists, collaborators all around the world, and also pharma companies, and you also have your own biotech company as well. If you could maybe highlight a few elements, steps, or happenings how could you set up all this huge network and way of bringing this back to the society?
Yeah, so I benefited throughout my career on having great collaborations, including, I didn't show it, the list of collaborations involved in getting the structures that I probably was awarded the Nobel Prize for. I didn't set out to necessarily, I need to get these collaborations. They sort of happened organically. Meeting people, being open with your data. I think that's really important. Being willing to share your data and not be afraid of being scooped. Gets access to a lot of other people with different technologies. But as I said, I didn't think I intentionally focused on this, but particularly involvement with chemists. So we've had two great collaborations. One is with Peter Gemeiner in Germany, and one is with Sasrota Majumdar. And I can't tell you how great it is to be able to, talk to the chemist and say, well, what would happen if we did this to this part of the compound? And they just say, oh, we'll let you know. In a couple of weeks, we have new drugs. And of course, being involved with people who can do the animal studies is also really important. But I don't have a recipe of how that happened. It just it seemed to happen.